SignalStar Multiplex IHC is a flexible and powerful tool for spatial biology profiling of tissues. If this is your first time using a SignalStar kit, if you need to troubleshoot the protocol, or if you simply want to know more about the protocol, this video is for you. I'm Derek, and I lead the Multiplex Assays team at Cell Signaling Technology. Our scientists developed and thoroughly validated the SignalStar protocol, and we continue to validate antibodies and expand the SignalStar catalog to broaden your experimental design possibilities. We also collaborate with technical support specialists and field application scientists to answer customer questions and offer protocol guidance for both manual and automated protocol users. Through our experience with SignalStar and interactions with the spatial biology community, we have found that walking customers through the protocol and highlighting key steps and aspects of the protocol improves their success rate. Numerous notes, cautions, and warnings are included throughout the protocol, but we understand it may be hard to prioritize all that information. In this video, we will highlight the most critical steps in a SignalStar experiment. With this knowledge, you can be confident that you'll get valid and reproducible results from your SignalStar kit. This video includes general tips that cover both manual and automated SignalStar protocols. Then, depending which protocol variation you will be following, we recommend also watching the section for manual or automated staining. If your SignalStar questions aren't addressed in this video or elsewhere in the SignalStar Resource Center, please contact us through this technical support link below this video or at cellsignal.com/support. This will connect you to specialists who will work with you to get your experiment running. We are here to help. So first, let's examine sample compatibility. To create SignalStar antibodies, we start with CST antibody clones that have been validated for IHC in formalin-fixed paraffin-embedded tissues and conjugate them for use in the SignalStar protocol. This means SignalStar antibodies will work on FFPE tissues. Other tissue preparations, such as flash frozen, can result in different epitope structures or availability and are not guaranteed to work with SignalStar kits. One of the most common protocol pitfalls that can lead to lower than expected fluorescence signal is incomplete mixing of buffers. Compare these two multiplex images of colon tissue. The staining on the left is a result of poor mixing of solutions, and the staining on the right was performed with end-to-end mixing for 20 minutes as our recommendations in our protocol, and this exhibits the expected signal intensity. This trend is quantified in this chart showing mean fluorescence intensity. Two of the kit components, SignalStar Antibody Diluent B and SignalStar Amplification Buffer B, are viscous. High viscosity reagents mix poorly unless gentle agitation such as mechanical rotation is applied for a sufficient period of time. Viscous liquids also tend to adhere to the sides of tubes or pipette tips, which can contribute to inaccurate volume measurement. To improve pipetting accuracy, use low retention pipette tips and pipette slowly. Keep your pipette tip submerged in the buffer for up to 30 seconds to ensure the volume is accurate and to avoid accidentally drawing up air bubbles into the pipette. When dispensing, also allow extra time to minimize retention of viscous liquid in the pipette tip and to confirm as much solution is transferred as possible. Pause at the bottom, then depress the barrel further. Use 15 mil conical tubes for combining components and incubate with end-over-end rotation for 20 minutes at room temperature to ensure complete mixing. Sometimes we are asked what to expect for signal brightness. SignalStar technology offers higher levels of amplification than traditional multiplexing techniques that use fluorophore-conjugated primary or secondary antibodies. If one of your targets is expressed at low levels, consider designing your panel so that low abundance targets are in the 594 or 647 channels. This helps avoid background autofluorescence that tends to occur in the 488 channel. SignalStar multiplexing can accommodate up to 8 targets in 2 imaging rounds. All antibodies are added in round 1. After the first imaging round, SignalStar oligos and fluorophores are removed enzymatically before amplifying the targets for round 2. To align the images acquired from round 1 with round 2, several commercially available imaging software packages and some open source options are available. Instructions for co-registration of images using the open source software QPath are available on our website. If you have questions, reach out and we'll be happy to help. As an optional quality control step, samples can be imaged after the fluorescence removal step. This can be used to verify complete removal of fluorophores from round 1. As long as the kit is used before its expiration date, we have found this control is not required, but it can give you extra peace of mind in the accuracy of your multiplex dataset. Ideally, samples should be imaged by fluorescent microscopy within 8 hours or as soon as possible after completing each staining round. Long-term storage is not recommended. Now that we've covered general tips for success, please review specific guidance for manual SignalStar protocol in the next section of this video, or skip ahead for specific tips for automated staining. Now we will cover additional tips for manual SignalStar, including when to prep materials and tips for antigen unmasking. A day before beginning the protocol, thaw these SignalStar kit components overnight at 4 degrees. Slide baking can be performed the day before as well. We recommend preparing solutions fresh before each round. This may be done during deparaffinization or antigen unmasking. Briefly spinning down the stock tubes containing antibodies or oligos for a few seconds can help to collect liquid that may be on the cap or walls of the tube before dispensing. If you need to pause the protocol to spread it out over multiple days, you can extend the imaging round 1 solution incubation containing the antibodies to overnight at 4 degrees Celsius. Once you have begun the amplification cycles in either round 1 or round 2, do not pause the workflow at any point. Antigen unmasking, also known as antigen retrieval, is performed prior to staining to make epitopes more accessible to antibodies. The SignalStar manual protocol includes antigen retrieval steps using either a pressure cooker or a microwave. Note that antigen unmasking is performed once, prior to round 1. Unmasking is not required for round 2. To optimize your microwave settings to maintain a sub-boiling temperature, refer to our Tech Tips video on this technique, how to perform antigen retrieval in IHC, by clicking the link in the corner of your screen. We have found that for some targets, the microwave method may yield lower SignalStar signal compared to pressure cookers, so results for the low abundance targets are not guaranteed. If desired, a control IHC staining using an unconjugated antibody and chromogenic detection may be performed. This can help to confirm signals seen in the SignalStar assay and to confirm or troubleshoot sample preparation and antigen unmasking steps. That covers our manual staining tips, but if you still need troubleshooting help, please contact us. Now, let's take a closer look at automated staining. Scientists on the Multiplex Assays team at CST validate SignalStar kits and products using the Leica Bond RX system. So, if you have access to this autostainer, you can follow the SignalStar protocol directly by selecting it in the Bond RX drop-down menu. If you don't see the SignalStar protocol there, be sure to check that the Bond software is updated to BXD40 or later. For other automated IHC systems, it may be possible to adapt this protocol. Please contact our team via the technical support link if you have questions about the automated protocol for your instrument. We recommend making reagents fresh and loading them onto the Bond RX once they are prepared. You can start the staining run immediately. The slides may sit after staining is completed, but we have found no effect on staining quality if they are imaged within 8 hours. Instead of transferring amplification solutions by pipette, pour the amplification solutions directly from the 15 ml conical tube into the Bond RX titration tube. Recap the tube and let it stand upright for a few minutes to allow the solution to collect at the bottom of the tube. Then pipette the remaining solution and add to the titration tube. Before running, check the reagent containers and refill in the Bond RX software as needed to ensure there will be adequate buffers and reagents. The Bond RX protocol includes a step for antigen retrieval in round 1. This is required only once before round 1, so in round 2, select blank in the menu. Thanks for watching. If you have any questions about the SignalStar protocol, kits, or imaging workflow, please get in touch with our scientists via cellsignal.com/support. This will connect you with specialists who can provide guidance to get your experiments up and running. Thank you.